Hello guys and welcome back to SH Academy. Sorry for the delay. So today we are going to take the lecture 7 and that is about the embryogenesis as part of plant tissue culture and their techniques. So last class we talked about the organogenesis. This is embryogenesis. So embryogenesis again it is nothing but the cultivation of plant cells only. So whatever we are discussing right now it's all part of this cultivation of plant cells. So different methodology, different strategy just to grow these plant cells. Okay, so the embryogenesis. Embryo is defined as the earliest recognizable multicellular stage of an individual that occurs before the development of characteristic organs of the given species. So in short, it means that from the callus, they are going to get the embryo. Okay. So the production of embryo-like structure from callus is known as the embryogenesis. In higher plants, such embryos usually arise from the zygote formation and are termed as zygotic embryos. So if you remember your botany classes, you know how exactly the fertilization or the zygotic formation takes place in the plant cells. Okay. So just go and brush up all these things so that it becomes easy for you guys to do the embryogenesis in the plant tissue culture part. So various types of cells are there and tissues, they can be used as a source of embryogenic cells. So it can be your microspores, zygotes, somatic cells. So any part can be used in order to carry out the embryogenesis. So from this, like from the previous lectures also, you can see that it's many a times, whatever we are discussing, you can see that any part of the plant can be used. It is the property of these plant cells having the totipotency that can be converted into the whole new plant. Okay, and this is only possible in the case of plant species only, but not in the other animal species. So embryogenesis can be induced in such cells by non-proliferative or proliferative direct embryogenesis, or by growing embryogenic callus, which in turn germinates and develops into the whole plant. So whenever we talk about this, it's all because of the controlled or the correct proportion of using phytohormones. Okay, then only it is possible. Now it can be initiated in an explant only from the more juvenile, that is very young, or the meristematic tissue. So guys, you need to remember this point because it's very important. If you want to carry out embryogenesis, you need to use the very younger part of the plant, or you can use the meristematic tissues. Okay. So immature zygotic embryos, cotyledons, hypocotyle dissected from the ungerminated seeds are common explants in order to carry out the embryogenesis. So isolated somatic cells can also develop into embryos, and it occurs through an organized sequence of cell division, enlargement and differentiation. So at this point of preparation for you guys, it's not required. So you can just remember that from the callus, you can grow into the embryo and you can carry out the embryogenesis. Okay. So the final stages of development towards maturation are distinguished by the overall enlargement and matured embryo morphology. So if you remember in the plant dev bio also, we study all these different stages of the zygote formation in the case of plants. Like you must have heard of heart shape, globular shape, torpedo shape, right? In the same way, in the callus or the plant tissue culture also you can study. So there are different steps are there. So first step is the induction. So induction is the initiative phase where the cells of callus are induced to divide and differentiate into groups of meristematic cells. Okay. So those are called embryogenic clumps. Important. Please remember. Now these embryonic clumps, they develop into initial stages of somatic. It is actually somatic embryo, that is globular stage. So the next stage is the maturation. So in this phase, somatic embryos, they develop into the mature embryos by differentiating from globular to heart shape. And this mature embryo then undergoes biochemical changes to acquire the hardness. Okay. And finally, it gets converted into to produce the seedlings or the seeds. Now let's talk about the advantages of embryogenesis. So the efficiency of this process is actually there is a reduction in the labor, of course the cost and the time. Okay, so that is the advantage of carrying out the embryogenesis. The potential for the production of much higher number of plantates and the morphological, cytological uniformity of the plantates. So it means that the number of plantates what you get after carrying out this embryogenesis is going to be high. Now, production of several plants on commercial scale includes, so it can be used for the food crops, vegetable spices, and mainly plant tissue cultures are grown or are done in mainly for the medicinal and the aromatic plants, and also for the ornamental plants also, like which is difficult to grow in the field. So for such cases, we use plant tissue culture. So that's all about the embryogenesis. So thank you guys for listening. If any doubts are there, please comment me. Or if you want any other explanation, you can also message me that. Happy to help. All the suggestions and the advices are welcome. If any doubts are there, please mail to us at Excelsior Academy 1228@gmail.com. You can also follow us in the Telegram channel by name Excelsior Academy. So thank you guys for listening to this. Stay tuned for the upcoming lectures so that we can complete this plant tissue culture series in the IIT JAM and GATB preparation.